Let's explore some keywords associated with DNA replication. Most of these are going to be strands and enzymes and other things that are important in understanding both bacteria and eukaryotic replication. So first we have the strands. So this parent strand and a daughter strand. This parent strand is the original. So here is the strand that you see in blue. This is the strands in the original molecule of DNA. It serves as the template strand. So once we start to pull this DNA apart, one strand serves as a template for one daughter, and the other strand serves as a template for the other daughter. So the daughter strands are the newly synthesized strand. It's also important to note here that parent and daughter can refer to the molecules. So this will be a parent molecule, or the original, and this would be a daughter molecule, or a newly synthesized molecule, okay? So we can have old and new strands, as well as old and new DNA molecules. The leading strand refers to the strand that is synthesized in the 5-3 manner. This is a strand that is continuously synthesized, okay? Versus the lagging strand that's discontinually replicated. The lagging strand goes in the opposite direction. So remember that our DNA is anti-parallel. So one runs 5-3 while the other runs 3-5. The one that runs 3-5 can't be synthesized continuously, so we have to synthesize it discontinuously using Okazaki fragments. So whereas the leading will have this perfect, neat strand, this one is going to have chunks of DNA called Okazaki fragments that are helping us to synthesize it. The primer is the short RNA sequence that DNA polymerase has to attach to. So DNA polymerase on its own cannot start to make a new strand. It can only elongate. And so what evolution has done was make this short RNA sequence that DNA polymerase comes, attaches to, and elongates, and then we'll go back and remove that RNA primer. Then you have these Okazaki fragments. So these are short DNA strands. So primers are short RNA strands, Okazaki fragments are short DNA strands, and these are the strands you find on the lagging strand that help us to synthesize it. Now, we talked about this previously, this origin of replication. So this origin of replication is where replication begins. This is where we open up our DNA and gain access to be able to build our strand. Once we open it up, it goes bidirectional. So we're going to have a replication fork running this way, as well as a replication fork running that way. So bidirectional is in both ways. The replication fork is where the parents separate and the daughters are synthesized. So here's our fork. So we're just going to keep opening up this molecule and synthesizing new strands. All right, let's talk enzymes. First, you have this helicase. So helicase's job is to unwind the DNA. So we know that our DNA exists in this twisted staircase. Helicase comes along, breaks those hydrogen bonds, and separates those nucleotides. And by doing so, we're opening up our DNA, making it accessible to rebuild the strands. We have single-stranded binding proteins. They are what their name implies. They're going to bind to the single strands of DNA, these parent molecules, and stop them from reforming the original molecule, okay? So single-stranded binding proteins comes in after helicase has opened up our DNA and stops it from reforming its double helix, stops it from going back to its native conformation. Topoisomerase 2, also known as DNA gyrase here, its job is to remove supercoils. So remember, we talked about this previously and we said it can introduce and relax them. So topoisomerase 2 is going to relax positive supercoils, which means it can introduce negative ones. And remember, we said that our DNA has this negative supercoiling that allows it to open up. So we're going to relax positive supercoils. This is topoisomerase 2, also called DNA gyrase. Ligase's job is to seal the sugar backbone. So remember, in the lagging strand, it's synthesized with these chunks of DNA called Okazaki fragments. Well, that's going to leave openings in the middle of it, and it's going to leave gaps. So ligase comes along and seals that up. So what kind of bond makes the sugar phosphate backbone? You should remember this. So phosphodiester, okay? Then we have primase, and it is, as its name implies, an enzyme that primes, right? Or the enzyme that synthesizes the primer. And what is the primer? The short RNA sequence that is necessary for polymerase to do its job. Okay? Polymerases. So in bacteria, we have DNA polymerase 3. Its job is to synthesize the leading and lagging strands of a bacteria chromosome. Whereas DNA polymerase 1, its job is to remove those RNA primers. We can't have RNA in our DNA molecule, so DNA polymerase 1 is going to get rid of those and fill in the gaps left once they take them out. And then ligase comes in and seals the backbone. Okay? A primosome is the complex between the helicase and the primase. Okay? So here's our primosome. So as DNA helicase is going along and unwinding, most specifically on the lagging strand, primase is adding primers as we go along so that polymerase can come along and create Okazaki fragments. A replisome is going to be helicase plus primase plus our 2 molecules of DNA polymerase 3. Okay, so this makes our replisome. Now, I want you to note here how our DNA is twisted and turned about. This is to help it be in the correct orientation to be able to function. So remember, we have a 5 to 3 and a 3 to 5. Our DNA can only read in one way. So we have to manipulate the DNA so that it appears to be in the correct notation. But because we can only do this with short areas at a time, we can only make these small strands called Okazaki fragments before we have to reconfigure it again. So in the meantime, here we have our single-stranded binding proteins stopping us from reforming. Okay, so primosome, helicase, plus a primase. A replisome, helicase, primase, 2 polymerases. And together, this replisome is replicating our DNA, opening it up, laying the primer, elongating for the nucleotides. Now when we turn our attention to eukaryotic cells, we have a pre-replication complex, or pre-RC. This is established in G1. Why G1? Because G1 precedes S when we have to replicate our DNA. So the first thing you do is lay down an ORC. So what does ORC stand for? Has anybody read anything in the chapter? Maybe, yes, no, of course not. So this is the origin recognition complex. This is the first initiator of our pre-RC. So this comes on. And then this says, hey guys, join me, and recruits CDC6, CDT1, and MCM helicase. And together, they're going to form the pre-replication complex. Now, once you get this MCM helicase, which is a helicase, and we know that the helicase's job is to open up, we get this DNA replication license. So once helicase comes aboard, it says, hey, this molecule is ready to be replicated. And this is especially important in eukaryotic cells because they have multiple origins. So the origins have to have this licensing in order for replication to occur. This is going to help us from replicating one area more than once. Okay, so this is a way that we can control replication, because remember too, we have euchromatin and heterochromatin regions. Heteros are more tightly wound up. We have to get in and undo them. So we're more likely to do the U areas first, but we need to know that, hey, this has been done, this hasn't been done. So they have this licensing that takes place. Now they also have a series of polymerases. So alpha initiates DNA replication. What does that mean? Excuse me. It starts it off. Alpha has primase activity, which means DNA polymerase alpha can create a primer. DNA polymerase delta synthesizes the lagging strand. So delta is responsible for creating Okazaki fragments. DNA polymerase epsilon synthesizes the leading strand. DNA polymerase gamma synthesizes or replicates mitochondria DNA. So if I say, where are you going to find DNA polymerase gamma? Nucleus or mitochondria? Your answer should be mitochondria. Then there's some special polymerases. So translesion replication polymerases, their job is to replicate damaged DNA. So we're going to talk about DNA repair and damage later on in this module. And so if there are some bulky lesions or thymine dimers, then there are special DNA polymerases that come along and replicate those areas. And we also have to note that DNA alpha, beta, delta, and epsilon are capable of repairing the DNA, okay? As well as a host of others, but these are what you should remember. So in addition to these polymerases being able to synthesize, they can also repair. Eukaryotes also have something called flap endonuclease, and it removes the RNA primer. So in bacteria, DNA polymerase 1 removes the RNA primer. In eukaryotes, flap endonuclease removes the RNA primer. And our eukaryotes also have something called telomerase. Telomerase is an enzyme that helps to elongate telomeres at the ends of the chromosome. So we'll talk about this. And telomerase is important, or not even it's important, but it's interesting because it has an RNA component, so it carries a template with it, as well as having reverse transcriptase activity. So we can take an RNA and create a DNA from it, okay? So telomerase is a special enzyme. So how do we feel? A lot of terms, but these terms, if you can start to read these and memorize them, will help you to understand the processes better.